want to do reference-based single-cell analysis with Azimuth on SciDAP. Azimuth is the name of the package that is written in R, and we just made a pipeline using that language. And feel free to interrupt me at any time you want to ask a question, because I think that will be kind of easier to make it kind of a conversation. So to begin with, I will briefly explain or remind what is single-cell technology. The main difference of single-cell sequencing is that in comparison with the bulk sequencing, we can track the origin of every sequenced read. And by origin, I mean the cell from where it came from. And for example, if we have a single-cell RNA sequencing, instead of getting the average expression, we can get the expression from every cell and then combine cells based on the similarities in its expression, get some clusters, assign cell types, and then proceed with analysis with a specific cell type. So, for example, compare cells within the cell type between different conditions. And there are several ways how we can track the origin of every sequenced fragment, and there are multiple technologies that allow us to do it. The two most common ones are droplet-based and well-based. Both of them share the same approach by using barcode in bead. Barcode in bead, we can see it on the left here. It basically provides information about the unique cell barcode and UMI, which is unique molecular identifier that is used for removing PCR duplicates. If we look at this part of the picture, this is what CellRanger usually uses, 10x Genomics usually uses, and that is a droplet-based. So we have every cell attached to barcode in bead, and then it's placed into the oil drop, and then that guarantees that all of the sequencing materials, oh, I need to show the that all of the sequencing material that we get from that cell will have this unique cell barcode. And another technology is well-based, and that's how the BD Rhapsody platform works. Instead of using droplets, they use a micro-well array, where every cell with barcode in bead is placed into a tiny well. And that's how they guarantee that all of the sequenced fragments will have this unique barcode. In both of these technologies, there is a situation when several cells can be attached to the same barcode in bead, and that is called doublet. There is a way to remove it computationally, but better to remove it before sequencing. And single-cell technology has its limitation as well. And one of the limitations of single-cell sequencing is that usually we don't have enough reads because the data is very sparse. And sometimes it's possible that certain genes just were not sequenced, covered by reads, and that we need to take into account when analyzing our data. Here I will show you what the basic analysis of single-cell data look like. It usually starts with the reads that we get from the, in this case, Illumina sequencer. Depending on the type of our single-cell data, we may have either 2 files or 5 or 3. In this case, I showed it for single-cell RNA-Seq, where the first read includes technical data, barcode and UMI, and the second read includes the actual sequenced fragment. Then with this raw read, unmapped reads, we run the software that will align them onto the reference genome. It could be either CellRanger, or it could be also a pipeline for a BD Rhapsody platform. But regardless of what we run, the output of most of these tools are saved in the form of feature barcode metrics, which includes cells as rows and features as columns. Features, in this case, it might be genes. If it was single-cell ATAC-Seq, it would be peaks. If it is multiome, we have several of such matrices, one for genes, one for peaks. And this data usually includes both low quality cells and high quality cells. As we can see here, it's unfiltered cells. And the next step is usually to remove all of the low quality cells. Again, depending on the type of our input data, we may apply different thresholds for RNA data, for ATAC data, or combined. Then once we have our high quality cells, we need to run dimensionality reduction, and visually it forms a UMAP from these filtered cells. And this step require certain parameters. One of them is dimensionality, how many dimensions to use. I will explain it in details on the next slides. And then once we have our UMAP, we run clustering analysis that will group cells by gene expression similarity if it was RNA-seq data. In order to run all of this analysis on SciDAP, we created a set of scripts that covers the most commonly used ways of analysis of single cell data. And then we put them inside Docker containers. Docker container, it's more like a virtual machine that guarantee reproducibility. So no matter when you rerun this workflow, if you have the same inputs and the same parameters, the results will be exactly the same. Even UMAP will look exactly the same in most of the workflows. There are a few that still may flip it sometimes upside down, or left or right. Then, in order to make our workflows portable and usable, but not only our platform, but by other platforms, we wrote the workflows in the CWL format. CWL stands for Common Workflow Language. It's just a specification that describes the order of execution of the steps, of the workflow steps. And it also explains which outputs of one step can be used as input for another step, which parameters can be configured, and so on. And all of these CWL workflows we run in the platform called SciDAP. SciDAP automatically can generate interactive user interface based on these workflows. It can also visualize results based on the outputs of these workflows. And it also allows us to chain different workflows and run them sequentially. Because all of this analysis, as I showed here, is split into logical parts. In order to make all of our workflows easy to understand, I created this scheme. There is a proper explanation of this scheme in this preprint that is available on bioRxiv. Generally, here we see each of these blocks is the workflow. The color indicates the type of the data with the which we can use for this workflow. If the workflow has two colors, that means it requires either both RNA and ATAC in this case, or it can be used for both RNA and ATAC-Seq data. So let's say we have our RNA-Seq data, so we select only green blocks. We start with our FASTQ files, then we need to map them. For that, we use Cell Ranger. Then if there are several samples, we need to aggregate the results, basically just merging the results from several samples. Then we need to remove low quality cells. For that, we use filtering pipeline. Then we need to reduce dimensionality, or in other words, to make a UMAP from these filtered cells. For that, we have this workflow. And then we want to group cells by gene expression similarities. And for that, we have clustering pipeline. The output of that pipeline can be used in the differential expression, in the trajectory analysis, differential abundance, which basically compares the number of cells rather than expression. That is the standard way how to analyze single cell data, but in these slides, I will show an alternative way. Let's say we already have the data set that we already analyzed. And we already assigned cell types for that, and we want to use the same cell types for a new data set. So here I will show you a sequence of steps that we need to do first to create the reference. that we will then use to map our query data set into reference. Sorry. For that, we had to develop several workflows. One of them is to load Rhapsody data, because the inputs for this particular experiment were sequenced on the Rhapsody platform. So we had to add the workflow to load the data sequenced on that platform. And then we created a new workflow that allows us to map our query data set into the reference data set. Both of the reference and query data sets. Someone keep calling me from Amazon. I don't know why. I can cancel it. So both of the reference and query data sets was prepared by Irina Rochman, and she showed it on her data club. I will skip the biological details, but briefly we just have 8 data sets that can be grouped by control, asthma, mRNA, mRNA viralimus, and then the new data sets belong to 3 groups: control, LNP, and mRNA. Let's see. So on the next slide, I will explain how we create the reference. For that, we need to run dimensionality reduction pipeline. Right, we have our raw data that we filter it, and then we need to run dimensionality reduction. I skipped the filtering step because it's kind of not related to this particular talk, but dimensionality reduction, it is related, because there are several parameters that are then used by Azimuth in reference mapping. So briefly, there are several big problems that we need to face when we run dimensionality reduction. One of them is that every cell might be sequenced with different sequencing depth, and that doesn't allow us to compare gene expression directly. Another problem is that we might have a group of genes that are highly expressed and group of genes that are not that highly expressed, and we need to scale them in order to be able to compare. And another problem is if we have several data sets, most probably we have some batch effect caused by time point, by donor, for example, or by sample. And usually before running this integration, dimensionality reduction and integration, actually as part of the dimensionality reduction and integration, we first need to remove these 3 problems. And in order to eliminate the differences between the sequencing depth, we apply normalization in order to eliminate the differences caused by the differences in the gene expression levels between the genes. We do scaling, and in order to remove batch effect, we do integration. Here we see an example of two samples. From each of them, we use only the genes that are highly variable, because in order to make it kind of easier for program to run the analysis, usually the program excludes the genes that are not variable within the data set. So this is just a visual representation. So let's say we run normalization, scaling, and then the next step is to run integration. There are several ways how we can run integration. The main goal of integration is to make sure that on the final UMAP, the cells that belong to the same cell type will be placed into the same area. So basically just removing the batch effect. And in this particular case, even if there are multiple ways how to integrate data sets, such as using Seurat integration, Harmony integration, or we can just even merge, for creating the reference for Azimuth, we need to run integration based on the Seurat. Well, integration made by Seurat. And how it works. Let's say we have 2 data sets. This is our cells, 1, 2, 3, 4, 5. 3 came from the first data set. Another 3 came from the second data set. We have 5 genes. First, we split it into 2 matrices. First data set, second data set. Then we apply a C-transform. It's a function from Seurat. It's it's kind of the most latest way how to normalize the data. So the way how it works, they first select the most variable genes within each of these matrices. And for this case, for example, gene E will be excluded and gene D from the second dataset will be excluded. Then they apply this function scTransform, which basically converts all of our read counts into what they call Pearson residuals, which shows us how the read counts are different from the expected mean for each of the cell, for each of the gene. And then I showed here that this gene is excluded and this gene is excluded from each of these two data sets. Then we need to somehow integrate both of them. And the way how Seurat does it, it needs to identify pairs of cells that can be used as anchors for integration. Or in other words, pairs of cells that eventually will belong to the same cell type. And for that, they use an approach that is called canonical correlation analysis. And this is kind of infographic where I try to visualize what actually happens. They try to identify those pair of cells that, based on a certain genes, can be grouped together. So in comparison to the standard PCA, because canonical correlation analysis is kind of similar to PCA, the difference is that we always run it with a pair of samples. So in this case, we have a pair of samples. We run this canonical correlation analysis for both of these data sets, identified groups of cells that can be used as anchors for integration. And then based on the expression of these anchors, we corrected our Pearson residuals that were first produced by scTransform and got the matrix that now can be used in PCA. And this matrix usually doesn't have any batch effect. It also includes the data that supposedly shouldn't include any of the results of the different sequencing depth or different scales for genes and so on. But this data that includes raw read counts will be used for differential expression. That's why in Seurat we usually keep this data and this data for different purposes. The next step, once we run this dimensionality, once we run integration, normalization and integration, we need to actually run PCA. PCA, it's an approach which allows us to remove noise from the data and kind of simplify it, because let's say we have 20,000 genes, and then after PCA we can get the same information shown in the only column with 50 columns, 50 dimensions, so maybe 30 dimensions. It depends on us and we can control it. And here I try to show an infographic that would explain how PCA works. Let's say there are two cells and their coordinates can be explained, described in the coordinate system where we have 1, 2, 3, 4, 12 genes. But most probably some of the genes, expression of some of the genes will correlate. So there is no need to save all 3 of them. And when we apply PCA, it will try to figure out which groups of genes can be replaced with a certain principal component. And that basically maximizes the distance between the cells. So here we see that these 3 genes were replaced with one principal component. These 3 genes were replaced with other principal component. These 3 with this. And this gene probably wasn't expressed at all, or it was expressed in the same way in both of the cells. That's why it was removed. Once we run this PCA, we need then to visualize it somehow, because 50 dimensions, it's still difficult for us to see as humans. At least for me, it's really difficult. So then we run another tool that is called UMAP projection, and it converts this PCA data into 2 dimensions. And from here, we visually can see how far the cells are, how they are grouped, and so on. But this doesn't answer yet if these 2 cells belong to the same cluster, if it's the same cell type. For that, we usually run clustering and cell type assignment analysis. We use our raw PCA data, and then following these steps, we can get our clusters, and find the gene markers, and assign cell types. In this case, it's manually. So the way how clustering works, we first use our PCA data, and these are the cells. And let's say we use 3 dimensions. Based on these 3 dimensions, we can see that cell 1, 2, 4 probably belong to the same cell type 3, 6, another cell type, because and if we see the big difference, the distance between this and this. But what if we use less number of dimensions? What if we use only principal component 1 and 2? So we look now at the projection of these cells, and now cell 3 and 5 are close to each other. That means, depending on how many dimensions we use, we will get different clusters. In this case, we use 3, so we have this cell type, and then some other cell, which might be also cell type. If we use 2, we would probably have 1, 2, 4 in one cell type, 3, 5, 6 in another cell type. Once we run our clustering and we identified our clusters, we need to find the gene markers. Gene markers are the genes that are upregulated in every cluster in comparison with all of the other clusters together. And based on these gene markers, we can manually assign cell types. And for that, we have a workflow which uses the output of the clustering pipeline and user-provided file with the names for each of the cluster. And here is an example of what I used as a reference for the next step of the analysis. So these are this is UMAP, which include 15 cell types that was analyzed by me for Irina Rochman's data. And we will use it as a reference. Now, how we work with the query dataset. Now we have, I already shown that we created the reference. Now we want to analyze the query datasets and map them into the reference. For that, we use data that we got from the Rhapsody platform. We have 9 samples. They all were sequenced within one cartridge. This is how I try to visualize the cartridge from BD Rhapsody platform. Each of these lane in the cartridge produced one RDS file. RDS file is our data file that was in which we stored Seurat object that was produced by this pipeline. This is the pipeline that BD Rhapsody provides to map their FASTQ files and create feature barcode matrices. So we got these 8 RDS files, and then for that we and in order to include them into SciDAP, we need to run this workflow, which will allow us to load the data into SciDAP. We select the workflow that is called single cell RNA-Seq BD Rhapsody import. Select all of these 8 files, upload them to SciDAP. Then we need also provide a file that describes the correspondence between the sample text and names of the sample. Sample text is what is included into the RDS file produced by this workflow. This is the sample metadata file. We also upload it into SciDAP. There is also another option to split additionally by dataset, but in this case we didn't need it. Then we click Save Sample, and this will add our Rhapsody platform data into SciDAP in a form that then we can use for subsequent steps of the analysis. The next step of the analysis is to remove low-quality cells. For that, we have the workflow that is called single-cell RNA-Seq filtering analysis. These are the thresholds that I used. We try to use very low thresholds in order to save as much cells as possible, because for this data, somehow the quality wasn't as good as we expected. And that's why we try to lower the thresholds to save as much as possible, as many as possible. So here I will show you how to use the output of the previous step as input for this pipeline. So we select the sample where we uploaded our Rhapsody data. Then we can additionally provide the file that shows how to group the samples, because we have 3 samples that came from the ASMA LNP, 3 from ASMA mRNA, and 3 from control. On this step, I show how to adjust the thresholds. Most of them, by default, already have reasonable thresholds. But as I mentioned before, for this data, I try to lower them. Each of the inputs has a short description of how it influenced the results. Once we adjusted all of the parameters, we click Save Sample, and then wait until the analysis finished running. Once it finished running, the pipeline produced multiple plots. This is just an example of some of the QC metrics that are shown in a form of a bilent plot for each of the data set. The same QC metric shown in the form of the scatter plot, where the horizontal axis in this case is RNA reads per cell, vertical is genes per cell. The color corresponds to the percentage of reads mapped to mitochondrial genes. All of these cross cells are doublets. Also, we can see the filtering threshold that were applied for each of the data set. Optionally, we can change thresholds per data set. And here, this is only one portion of cells that are used, that we assumed are high quality cells. Everything else will be removed. And the same data can be shown in the form of the density plot. For this particular sample, these are our thresholds, so we used only this portion of cells. Then, when the workflow finished running, we can see the results for unfiltered data, all of the QC metrics before we applied filters. The same QC metrics split by group and condition, because you remember we have 9 samples grouped into 3 groups. The same QC metrics for filtered data. Some of the plots are a bit redundant, but we never know which one are useful, so I just output all the possible QC metrics that I was able to calculate. Also, all of the outputs can be downloaded in a form of the file. We also produce PDF plots and interactive visualization. This is not UMAP, this is just a QC metric where the horizontal axis is reads per cell, vertical axis is genes per cell, and we can see our cells where they are placed. We can color by condition, by data set, by reads per cell, genes per cell, mitochondrial percentage, and so on. Also, every workflow produces a log in the form of the HTML file where you can see all of the code that was run and all of the results in a form of plots that are embedded in this report. So you know where exactly that plot was created and by which portion of the code. And on the left, this is just an example of how to use filtered QC plots for one particular sample. For example, on this plot, we see reads per cell distribution, and we're interested where these cells came from, where they are on the other plots. And we see that this corresponds approximately to 400 reads per cell. So we can go to this plot and see that this is also reads per cell. 400 reads per cell correspond to this number of genes per cell. This number of genes per cell is here on this density plot. And on this plot, we can see the mitochondrial percentage and reads per cell, and approximately know what was the percentage of mitochondrial genes for these cells, for all of this. I hope one day I'll be able to do it interactively, but for now, it's just in the form of the static plots. So once we run the analysis, we have all of our cells, only high quality cells. And then the next step is to actually map this high quality cells into the reference. On this slide, I tried to put some infographic that would explain how the reference mapping works. On the left, we see our query data set. All of these cells may come from one sample or from multiple samples. In comparison to what we usually have in the dimensionality reduction, where we split by sample, here we apply a C-transform for all of the cells together. But the difference is that we need to normalize our cells in exactly the same way how the reference was normalized. For that, from the reference data set, the program used this thing. It's called SCT model. It's basically a set of parameters that is then applied for our row read counts to make our reference data set and query data set comparable. And another thing that we get from the reference data set is the set of the most variable genes, because as I mentioned before, we don't need to use, or if I didn't mention it before, we don't usually need to use all of the 20,000 genes because it's computationally heavy to run. So all of these tools often select top 3,000 or 2,000, the most variable genes. And in this case, when we run the analysis to create the reference, we have some ABC most variable features. So when we want to normalize our query data set, we want to use exactly the same features. Again, because we want to compare them. So that's why from all of these genes, we use only ABC gene, and these are excluded. The next piece of information that the workflow takes from the reference is the assigned cell types. And in order to map these assigned cell types into already normalized data, once we apply this AC transform, we have normalized data. We use two functions that are part of the Seurat package that basically finds, it basically kind of integrate our query data set and reference. Similarly, how it was between the datasets, it also finds some integration anchors, and then based on those integration anchors, we transfer the labels. And these integration anchors are usually taken from the PCA of our reference dataset. And the next step is to make a UMAP that would look the same as the UMAP of our reference dataset. And for that, they use also a set of parameters that define the shape of the cells, the shape of the UMAPs from our reference dataset. So 1, 2, 3, 4 pieces of information are important to map our query dataset into the reference. And I also put the links, some information where we can find more information, more details about how this reference mapping works. So this is an example of the workflow, how we run this workflow. We need to select the sample that will be used as a reference. So this sample should be analyzed on SciDAP. The reference data set can be selected either from SciDAP, or we can manually upload files that can be downloaded from this website. And I will show in the video what is this website. And then there are two QC thresholds. Both of them define how reliable are the results of the reference mapping. The first one is called prediction score, and it indicates whether the cell type assignment was accurate. And the other QC is mapping score, and it shows whether the projection of the cell from the query dataset was accurate into the reference UMAP. Or in other words, it shows whether the cell from the query dataset is represented by the reference, because sometimes we might have cell types in our query dataset that are not present in the reference. So on this video, I will show you how to run this analysis on SciDAP. We type the name of the analysis, we select our query dataset, which in our case, results of the filtering analysis. Then we select the sample that will be used as a reference. Alternatively, we can upload two files that can be also used as a reference. And those two files can be downloaded from the Azimuth website, which I will show you in a few seconds. So this is the website where we can download the references. They created, I think, approximately 10 or 15 references with a detailed explanation based on which data that actually created it, which cell types are present, and they also provide the link where we can download two files that can be used as a reference. One RDS file, another is index file that is also used by Azimuth. Then here we provide two thresholds for the prediction confidence score and prediction mapping score. By default, I use 0.75, what was suggested by Azimuth. For all of the predicted cell types, we can also identify gene markers, bit markers, if it is run for multi-omic data. We can export results into HTML report. And click Save Sample. This is some of the results that we have from running this analysis. On the left, we see our reference data set. On the right, this is our query data set that includes only cells that passed our QC thresholds, which was 0.75 for prediction score and 0.75 for mapping score. Actually, this is confidence score, this is prediction. So out of 26,000 cells, we got 13,000 that passed QC, and we see that the cell types more or less correspond to what we had in our reference data set. And in this case, the reference data set had only 11,000 cells. If you have a reference with a bigger number of cells, it's even better. This is another example of the outputs that we can get from our pipeline. These are two of the query data sets that includes all the query cells, not filtered by the QC metric, but they're colored by this QC metric. On the left, it's a prediction score, so we can see which cells passed this threshold for the prediction score. On the right, the same UMAP, but colored by the prediction mapping score. And here I put some short explanation of what the mapping score and what is the confidence score, what they actually explain and how it is calculated. So even in few words, the mapping score, in order to get this mapping score, they first map cells from the query to the reference, and then from reference to the query, and see kind of some metrics whether the cell from the query dataset is well represented by the reference. And the confidence score, the cells that are in between the clusters, we don't see them here, but there are some small cells here. They have very low confidence score because they are in between the clusters. This is another output. We can see what was the percentage of cells from the query dataset that passed QC. And on the right, this is a UMAP colored by cells. Green are those that passed QC, red are those that failed prediction QC, and these are the cells that were excluded because of the low confidence score. These are the cells that somehow got excluded because of the low mapping score. And if someone is interested what it would look like if we map reference on the reference, that's what we get if we map our reference dataset on itself, just kind of a safety check to make sure that the workflow works. But there are still some cells that didn't pass QC, which is weird, but well, probably because there are some limitations. So if we come back to our query dataset mapped on the reference, on the left we see our UMAP, which includes only high quality cells colored by the dataset, and on the right the same UMAP colored by the group and condition. The reason why I put these two UMAPs, just to show that even if we didn't run integration for our samples, we still see that the batch effect caused by sample was removed, but the biologically meaningful information is still present because, as far as I understand, control and asthma LNP should overlap in certain degree, and asthma mRNA shouldn't. And that's what we see on this UMAP. So it was like a visual test whether the data sets were well integrated or not. Another example is for all of our cells, we can build QC plots. In this case, we have reads per cell and genes per cell for each of our cluster, colored by the prediction score, prediction mapping score, and on the left by the prediction confidence score. The only reason why save these plots because maybe we will be able to see some correspondence between the low quality cells and low prediction or low mapping score. In this case, I couldn't find any, but just in case I save those plots. And another way how to check the results was to build the heat map of the top gene markers that we have from the reference data set and use the same gene markers and build the heat map using the query data set, including only those cells that passed our QC. So as we see on the left and on the right, the heat map mostly correspond. That means that the cell types that we assign based on our query data, on our reference data set, more or less reasonable. The only difference is the number of cells. So, for example, here we don't have much cells, so we don't have any of the highlighted cells here. And another feature that we added not only to the reference mapping pipeline, but into all of our clustering pipelines. We have clustering for RNA, clustering for WNN, and cell type assignment. It's calculating of gene set model scores. This is a standard function from Seurat. The idea of this model score is to show the average expression of the group of genes. You can either provide this file manually, where you have a gene set name and the list of genes that you want to calculate average expression for, or you can download this file from MSeqDB. This is the database that is used for gene set enrichment analysis. And in this particular case, I downloaded the file that includes all of the hallmark gene sets that represent well-defined biological states and processes. And on this dot plot, we see every of the gene set names. The color indicates the average expression. The size shows the percentage of cells where the gene set was expressed. On the left, it's just an example of 3 UMaps, which I thought might be interesting to show because they were the most contrast. I actually had 50 UMaps, so I just manually selected 3 of them. So we see the area of cells where this average expression score was high, and it corresponded to this gene set name. There are also gene sets where they have different cell types, so you can use it to help to assign cell types also. And this is an example of the outputs of this pipeline. So we see our reference UMap, we see all of the QC metrics. On the left, everything is related to the confidence score. On the right, everything related to the mapping score. Then we have our projected UMap. We have the proportion of cells that came from each cell type, from each of the data set. We have our gene set of interest. We have a heat map of gene markers. The same gene markers saved in the table, which you can also download as a file. All the files also can be downloaded directly. You can download even RDS file and then continue analysis manually. Also, we have a cell browser where you have UMap with the assigned predicted cell types. You can color by the prediction mapping score, prediction confidence score, change the scales, the color maps. We can also filter cells that passed prediction QC. In this case, we select only true and then only show selected. So now we have only cells that passed QC. For each of these cells, we can click on the name of the cluster and see the gene markers. We can click on the gene marker and see an expression of the gene. We can also manually type the name of the gene. Color by dataset, condition, all of the filtering QC metrics. Also, because we provided the gene set file, we can also see the average expression of each of the gene sets that was in that file. So on the left, you have the names of each of the gene sets. You can click and see average expression. And you can see the HTML log which includes all of the code that was run and all of the plots placed in the place where the corresponding code was executed. It's easy for debugging and also easy to rerun some part if needed. But there are some limitations of this approach. So the first limitation is that Azimuth as a tool is not optimized to discover novel cell types. So if they're not present in your reference data set, you cannot map it. Most probably, your cells will be mapped to the closest cell type. Another disadvantage is the level of granularity. That means if, let's say, you have a cell type that would usually include several subcell types, but it's called as one cell type, you won't be able to separate them in your query dataset. And I will show example on the next slide. And another, it's not a disadvantage, it's just a thing that we should take into account. Both of your reference and query datasets should be mapped on the same annotation, genome annotation, because you might have some genes that were renamed, and then it will impact the results dramatically. And as an example of this limitation, that is called the level of granularity in cell type annotations depends on the resolution of your reference dataset, I reanalyzed the query dataset using the standard approach, just running the dimensionality reduction and then clustering, and then try to find the correspondence between the results that I get from the reference mapping and the standard approach. And here we see that there are two cell types that by Azimuth were assigned to one B cell type, but in fact there are two clusters. And here they are even separate on UMAP. And when I looked at the gene markers, I could see that most probably both of them are B cells, but they might be slightly different B cells. And I cannot judge what are these subtypes of B cells, because for that I need more biological knowledge. But based on the Google and ChatGPT, it showed me that they're both different stages of B cells. And that was it about the reference mapping. And many thanks to all of the people listed here, especially to Mark and Irina Rothman for guiding me through biological interpretation of all of the data that we received. And if you have any questions, I will be happy to answer.
Is there any QC to make sure that the Azimuth does not force one cell type on another? Because, like, for example, sometimes there is, especially like in myeloid cells, there is multiple subtypes, and then does it only give like the general definition? If you say this is, for example, this one has macrophages, and then there is subtypes. So does it force it into the macrophages, or?
It will probably map it to the closest cell type, but for those cells, as far as I understood, the mapping score will be low, because usually the mapping score indicates if the cell type from the query dataset is not represented by the reference. But if you have these several subtypes represented by the reference, but they are called as one big cell type, then you will not be able to distinguish them in the query dataset. They will be mapped to that one big cell type, and they will have the high mapping score. One thing that I didn't include in my analysis, there are another two QC metrics that in general say whether the query dataset and reference dataset similar to each other, and they calculated based on the number of the integration anchors, which are cells that share the same biological state or gene expression. But I don't output it here in my pipeline because I wasn't able to do it quick enough. So maybe later on the next iteration of this workflow, I will add that metric as well, and it will tell you in general if you can use this reference with your query data set.
And what about unique cell population that are in the tiny portions that you want to identify in your new data set? Like, you know that they are present in the original data set, it's present there, and then you want to see if in the new data set they are also present. Will it be able to identify them?
I think in this case, yes. As long as they are present in the reference data set, I assume Azimuth will be able to map them on that cell type.
And we saw it's called. You have to
Yeah,
make sure that you have everything labeled.
So even if, so it has to be a unique small subpopulation that defined as a cluster in order to put that definition on them. It cannot be mingled in other populations. Okay, so it cannot say these cells among this big population are the one that you're searching for. It cannot do that.
No, it cannot.
So from the beginning, you need to find the right number of clusters in order to find them.
Yes.
So this is a bit tricky. So it's nice as long as your data set, you know what you're looking for,
Right.
and you know exactly how to create the UMAP to make sure that the UMAP from the beginning will contain the subpopulation that you're interested. You just want a validation to show that this is specific, the subpopulation that you're searching for.
Right. So I look at this workflow as something that allows to save some time, but it doesn't mean that you don't need to run the standard analysis by dimensionality reduction and clustering. But you can also use it as a comparison. You have your old data, you want to get some preliminary results, you run this reference-based mapping, you get some cell types, then you see, oh, this part is interesting, but I want to see in details what is there. Then you can either subset to that cell type and run the standard analysis, dimensionality reduction and clustering. Or maybe you can just run in parallel the standard approach and then compare. Because this reference-based mapping can be run on any stage. I just showed that I run it after filtering, but you can do all of the other regular workflows, and then after that run reference mapping and compare your UMAP with the one that you got from the reference mapping. And another detail is that on the website where they provide already made reference models, they usually have several levels of cell type names. They have kind of top level cell types, then more detailed and more detailed.
Yes,
and depending on what you need, you can choose which one you want to use for your reference mapping.
Thank you.
You're only as good as your reference model, though.
Exactly.
A caveat to the Azimuth is that it doesn't have some populations that are standard to blood.
In
Like
the models that they provide.
NKT cells are not listed. So if you wanted to map just blood, those get redistributed to different spots and have low mapping scores. So
Yeah.
if you're I mean, it's good for an overview. And if you have a really good reference for yourself, you can use that if you have really great labels.
So for the Rothenberg that are interested in the esophagus, theoretically we can take the big data set that is present that they created on the esophagus from Aviv Regev, the late one, and basically put it as the azimuth. There is a
Atlas?
Yes. Can we do that?
We can, but for that you need no, it will work. But you need to be sure that the cell types in that EOE atlas are good enough.
They are very specific. Regarding good enough, yeah, I agree with you, especially specifically on the eosinophils that have only one genes that, and I can tell you that eosinophils express CLC. But yes, I agree with you. But for like, for example, if I'm not familiar well enough with a specific subpopulation and they already identify them, I can use their identification, put it on mine, and then I will be able to, okay, now I want to look at my data set.
Yeah, you can do it.
So that's pretty cool. Thank you.
One thing to keep in mind is that if they're mapped on certain version of reference genome and you use another version, and we had that problem with your data once,
Yes.
the gene name should correspond, especially if they are renamed.
Oh, yes, that's for sure. Thanks. That's cool.
Welcome.